Hi class, this is Sir Alex Basco and welcome to our lesson in General Biology 2. Today we are going to discuss something new. It's a little bit related with still the genes and everything, but today we are tackling basically about genetic engineering. There's a lot of things about genetic engineering and you could actually take years studying genetic engineering. So today we're just going to tackle a little bit about it. Basic stuff, nothing really deep. But we're gonna learn about what genetic engineering is, the implications, the history behind its development. So that's everything that we're going to talk about today. So let's start. But first, let's understand why do we want to study this. So the goal of today's lessons is for you guys to be able to outline the process involved in genetic engineering. Again, we're just going to deal with some very basic processes because it can actually take advanced understanding and knowledge of genes in order to fully understand what genetic engineering is and apply it to everyday lives or in a lot of things that we use and do in our everyday lives. But today we're just gonna be a little more informed on what genetic engineering is. Next, we're also going to discuss the applications of recombinant DNA or recombinant DNA, what recombinant DNA is, and where do we apply them. So let's start with genetic engineering. What is genetic engineering? Simply said, genetic engineering is basically the manipulation, modification, and recombination of DNA. So if you guys remember our discussion about DNA, DNA is basically the blueprint of life, as we say. It's because of the central dogma of biology. DNA contains the information that the body needs in order to create proteins, and proteins are necessary in order for us to first function. A lot of the things that happens in our body is because of the proteins, and a lot of the physical characteristics that we have is this is because of the proteins that were encoded and transcribed and created from DNA and RNA. Without these proteins, a lot of our physical traits wouldn't manifest. We won't be able to manifest as male, as female. We wouldn't know if we're tall or if we are short, because that's still part of the genetic, that's part of our DNA. Of course, there's various factors on height, but one of the factors that affects height is actually the genes. Also, whether our hair is straight, is it wavy, is it curly, do we have dark eyes, do we have brown eyes, that sort of things. All of those are expressed because of our DNA, because our body encoded, transcribed and copied the information is stored in our DNA and expressed it in our body. So DNA has been a very interesting subject in biology because what we can do now with DNA is we can actually manipulate it, modify our DNA, and create various combinations of DNA to express very specific traits that we want. So that's what we call genetic engineering. And genetic engineering is a general term that we use to define various methods that we do to create what we call a recombinant DNA technology. Recombinant DNA technology is basically taking two DNAs from two different species and putting them together. More specifically, one of the things that we do is take the DNA of a human and DNA of a bacteria and actually put those two together and create a various what do we call this, and we want to duplicate those combined DNA. If you guys remember your DNA duplication process, Sir Aman discussed that. So that's what we want to do. We combine two separate DNA from two very different species, and we want to replicate those combined DNA and see what we can do about them. So that's nowadays, that's what we talk about when we talk about genetic engineering. Basically, what we call recombinant DNA technology. And we're going to discuss more what recombinant DNA technology is later on. But why is this necessary? How can we actually use this recombinant DNA technology? Well, we've been actually using it A lot of the techniques that have come from recombinant DNA technology have brought us one production of medically important products. As an example, because of recombinant DNA, we were able to create human insulin. And insulin is very important, especially for people that are experiencing, diabetes. More specifically, or more importantly, for those who has type 1 diabetes. Type 1 diabetes is different from type 2 diabetes. Type 1 diabetes are people that have a genetic, it's an autoimmune disease, meaning it's a problem that was hereditary. They got it from their parents and it expressed. It's an issue of a genetic problem in our body that our body is targeting I believe it was the HEPA cells in our liver, and that the body cannot create insulin. That's the issue for type 1 diabetes. And insulin is necessary in order for glucose to go inside the cells. If you guys remember your glucose, we need that glucose to create ATPs. And if those glucose are not going inside the cell, the body cannot make ATPs. Hence, people with type 1 diabetes have actually difficulty having the cells, take up that glucose. So what we do is to create artificial insulin that the body usually make, and they inject it to themselves, so that they can have the insulin that they need so that their cells can take up that glucose and put it in their cells so they can create ATPs that they can use for a lot of their energetically needed processes like breathing, moving, blinking, and basically living. So that's the issue with type 1 diabetes. Type 2 diabetes is more of the lifestyle. So those people that are have issue, that are usually, most of them are obese or have issue with not moving enough or overeating. That's type 2 diabetes. But type 1 diabetes is something that is hereditary, and one of the treatment that we need is actually insulin. And we can create insulin now via what we call recombinant DNA technology. Another important development from recombinant DNA technology is what we call the GMOs, or genetically modified organisms. So some of you may have been, if you're using TikTok or social media, there's a lot of what we call misinformation about genetically modified organisms, or the GMO. A lot of you may have heard that GMO is really pretty bad. Of course, up to this day, there's what do you call this? Um, it's an ethical issue, because later we're going to discuss about ethical issue, but not all genetically modified organisms are pretty bad. Some of the things that we have done, for example, this is a salmon, a genetically modified salmon. A normal salmon usually take about 3 years to grow. So, um, and since we consume salmon, and more people are consuming salmon, it's a food product. One of the things that we want to do is to, for us to be able to increase the production of food. So one of the things that they have done is via recombinant DNA technology is have salmon be able to keep producing growth hormones. So what happens is that instead of having smaller salmon, we have bigger salmon, and bigger salmon means there's more meat, there's more food that we can use. There's more food production. And instead of growing 3 years, we can now grow salmon in just about 13 or 18 months. So see, like we cut it from having 3 years to 18 months. So that's like a year and a half. We cut our production of salmon from 3 years to a year and a half. So we have more food product. So that's one of the things that are happening via the use of recombinant DNA technology, not only in salmon. Some of the things that we use recombinant DNA technology is to create crops. Crops are usually plants that we use for food, such as wheat, barley, rice. Those are crops. And we can create uh, insecticide type of plants, meaning we do not need to use insecticide to kill the insects that could eat our plants. They could resist those insecticides, so our plants are not get damaged and they won't get eaten by a lot of um, insects that may harm our crops. So that's one of the things that we can do with recombinant DNA technology. One of the things that we have done with recombinant DNA technology, if you've seen in the US, or I've been in the US, our onions here compared to the onions in US are very different. You can think of, if you have ever held a baseball, so that baseball, it's actually quite big. It's if you make a fist with your hand, maybe two fists of hands. That's how big their onion is. And they managed to do that via recombinant DNA technology. Another thing that we have done for food production with our genetically modified engineering is seedless watermelon. If you guys like watermelon, sometimes those seeds are, they're not an issue if you eat them, but sometimes you just want to keep eating. You don't want to keep sprouting or removing seeds as you eat. So one of the things that we have done is seedless watermelon. So not only are we changing them and making them bigger for us to produce more food, but also one of the things that we have done is to with our food is to increase the nutritional value of food. So that's a lot of the application for genetic engineering, more specifically recombinant DNA technology. So a lot of good things. But later on, we're going to discuss some of the issues about genetic engineering. And of course, I think by now you understand why there are some issues. But again, so just to clarify about misinformation about genetically modified organisms, GMO it's just labeling, especially on food. Uh, a lot of them are actually not really are not bad in our body. In fact, some of them are actually more nutritious for our body. But yeah, so please do not be misinformed about GMOs or genetically modified organisms like our food. Continuing on, let's now discuss what is recombinant DNA. So when we talk about today, when we talk about genetic engineering, we are more specifically talking about recombinant DNA technology. So what do we do during the recombinant DNA technology? So what we do is, well, we use a restriction enzyme, or specifically a type 2 restriction enzyme. And this restriction enzyme, what it does is it actually cuts the plasmid. If you guys remember plasmids, plasmids are DNA rings that is present inside a bacteria. If you guys forgot about plasmids, I want you guys to go back to our biology one. I believe it's in lesson 2 or lesson 3, somewhere there. So plasmids are rings of DNA that is found inside a bacteria. It has it's a DNA ring that is not the main. For example, if this is our bacteria, that's the DNA of the bacteria. Then they have plasmids that also has DNA in them. Okay, so you can think of this as our the DNA in our nucleus, and the plasmid is the DNA found in our mitochondria. So you can think of that. So the plasmids are basically rings of DNA found inside a bacteria. And what the restriction enzyme does is it cuts and opens these rings. So as you can see here, it managed to cut. But if, for example, if we use a donor DNA, like for example in humans, what it does is it actually cut it in multiple places. And using a restriction type 2 restriction enzyme, we can actually very specifically identify which cut we want to make, which so you can which section do we want to cleave. Now we can do that nowadays with a type 2 restriction enzyme. So that's what it does. First, we introduce a restriction enzyme to a DNA. Donor DNA could be from a human, an animal, or a plant, and then introduce it also to the plasmid of the bacteria. And then the plasmid of the bacteria gets open, and the DNA of the donor gets to splice into 1,000 sites. So you get these smaller pieces that are very, very specific. And then what it does is we actually combine those two together. You can see it here. So we can combine those two sections together. So from this cut up site from the donor DNA and the open site of the plasmid, you can attach them together, and using a DNA ligase. If you guys remember your DNA ligase, it's the one closing your DNA. DNA ligase can seal those two together. And what we create is what we call a recombinant DNA plasmid. So we have a combination now of a DNA from the plasmid of a bacteria and the DNA from our donor. It could be a human, animal, or plant. So the DNA ligase seal those combination. So now we have created a recombinant DNA. And what we're going to do next is we want to introduce this recombinant DNA back into the bacterial cells. And if the conditions are right, the bacteria would actually accept this plasmid. Sometimes it will reject it. And and then we just have to find the correct bacteria under certain correct conditions. And if we successfully manage to introduce the plasmid, this recombinant DNA plasmid into bacteria, what the bacteria does is what it will do is binary fission. Because forget what binary fission is, it's basically the duplication process of bacteria. For us, we have meiosis and mitosis. For bacteria, they have binary fission. And when they do the binary fission, what happens is they will start to duplicate also this recombinant DNA plasmid. And now what we have is multiple copies of that recombinant DNA. And more specifically, there would be, since we have a lot, now we can identify, or what's more interesting is that the DNA can actually express the proteins that is encoded in this section. Remember when we were creating proteins, we are reading a section of a DNA to create the proteins. I hope you guys remember this. The protein synthesis, and the bacteria can actually also create that proteins. They can create, and when they express that proteins, then we can identify, we can apply it to a lot of the things that we did. Now we can make, for example, insecticide resistant plant. We can have this protein to be a growth hormone. Now we can introduce this, what do you call this? We can now use this to start creating growth hormones and put it in a salmon, and then the salmon now grows much bigger and much faster than we do. So that's what we call genetic engineering. That's what recombinant DNA technology is. This is the process on how we do genetic engineering. So I hope it's not confusing. If you guys have any question about the process, do not hesitate to message me. So that's what genetic engineering is all about. So why is it very important? Well, recombinant DNA technology is used to create and produce new genetic combinations now that we can use in science, medicine, agriculture, and various industry. It has a lot of uses. But the fundamental goal of recombinant DNA technology is for us to be able to isolate, characterize, and manipulate gene. Isolate that gene, identify what that specific gene does, and manipulate it in some way in order for them to be useful in science, in medicine, in our crops, in our food, and in various industries. Another very recent development of genetic engineering is what we call the CRISPR-Cas9. So the CRISPR-Cas9 is a very sophisticated gene editing tool. And what it does, it actually allows researchers to alter DNA sequence and modify gene function. What, what we mean by that is if we have a DNA, we can isolate a specific part of that DNA, especially if that DNA, let's say, is a disease causing DNA. So, for example, of this broken DNA is color blindness. Color blindness usually happens in a problem in the X chromosome. Not a lot of women has color blindness because if one of their X has issue with the color blindness, the other X can produce the protein necessary in order for them not to be colorblind. But that's the issue for men. If men's X chromosome contains that specific issue in the X chromosome, we do not have another X chromosome because what we have is a Y chromosome for men. And what happens is that no chromosome, or we don't have that extra X chromosome to produce the protein necessary in order for us not to be colorblind. So that's one of the issue that we can do. We can use CRISPR Cas9. We can take that colorblind person's X chromosome, specifically a male, and then we can take out that broken DNA section and then alter it and make it not broken DNA. That's one of the things that we can actually trying to improve now, a CRISPR, and we call it CRISPR Cas9. Another thing that CRISPR Cas9 that can be very valuable is using it with people with HIV. HIV is still an issue because we actually do not have a cure for HIV. What we have is to prevent the production of the protein that causes HIV. It's a whole lot of complicated process, but we can use CRISPR-Cas9 to, if we improve this, we can actually finally cure HIV, not only prevent it. So something like that. So that's one of the things that we can do now. It's a CRISPR is a gene editing tool. So the potential of CRISPR, one, like discussed, correct genetic defects. We can now treat and prevent the spread of diseases. So a lot of the diseases that we have is caused by, issues with our DNA. That's why we do newborn screening. In newborn screening, they can identify the genes that the baby has that could potentially be, an issue for them, and informing the parents on what they can do to prevent that happening to their children. Now that we can, if we improve on CRISPR-Cas9, we can edit that gene so that the disease would not manifest. We can improve growth and resilience of our crops, and this is the one that I'm mentioning about. This is called gene therapy. This is where we introduce a normal gene into the genome to repair the mutated genes. So for example, we have a mutated gene, like the example that I've given in male X chromosome, which causes them to be colorblind. We can take that out and replace it with a normal functioning one so that the baby male will not be colorblind. Okay, so that's one of the biggest potential of CRISPR and recombinant DNA technology. I hope it's clear. If you guys have any question, just message me. Now let's learn about the historical development of recombinant DNA. First, in the beginning, when we talk about genetic engineering, or now when we talk about genetic engineering, you may be thinking, wow, this is very advanced. This is something new. But in reality, this is actually genetic engineering is not something new. We have been doing it for quite some time now. Although it's not as invasive how we do it nowadays, it's not really as much as advanced that we do it today, but we have been doing it. We have been, trying to manipulate the genetic makeup of organism via what we call hereditary and reproduction. Some of the things that we have done is, for example, artificial insemination, in vitro fertilization or test tube babies, and even cloning. So what do we mean by this? So if you guys remember Mendel, Gregor Mendel is the father of genetics. So he's a priest, I believe he's a parish priest that discovered that if he has, he can actually manipulate the plants. So if he combined two plants with different colors, what would be the color? Would it be the color of the other plant, or it be the color of the other plant, or will it be a combination of the color? So, or for example, if you have two peas that are short, if you combine it with a peas like the green peas, that is tall, if you combine them together and let them reproduce via hereditary reproduction, would the offspring peas would they be short, or would they be tall or long peas? No? And that's a type of manipulating the genetic makeup of organisms already. And we've been doing that with a lot of our animals and our plants. One of the things that we do is artificial insemination. wherein if we have, for example, an animal that is much bigger, we take the semen of that male animal and then use that to put it inside a female animal. We usually do this, for example, in a lot of our food production, like in pigs. I've seen someone does an artificial insemination wherein they take a male pig that is much bigger so that the female pig, when it get inseminated, you're basically inserting semen into, the vulva or into the ovaries of the male pig to get them impregnated without them having copulation or sex. So that's what artificial insemination is, so that the female can have babies. So we can specifically choose which male animal we want to have, specifically if this male animal have a characteristic that we want to manifest in its offspring. So another thing that we do is in vitro fertilization or test tube babies. Do not get confused what do we mean by test tube babies. Are we developing babies inside a test tube? Not specifically. In vitro fertilization is basically taking the semen, for example, especially if, for example, if the woman has a type of autoimmune disease, what we can do is we take the egg cell of that female with autoimmune disease, and from the various samples of the egg cells that we have, we can identify which of the egg cells um, contains what we call the autoimmune genetic, so the genes that contains the autoimmune disease, and then we can choose a healthy egg cell. So we can choose an egg cell that does not have that autoimmune disease, and what we can do is we can specifically choose that egg cell and then use that uh, and in a test tube. Basically, you can just imagine this: we place that egg cell inside a test tube and then place the father's sperm cell inside the test tube and then let the zygote create, and then we put the zygote back into the female, so let the female uh, let the female give naturally develop the baby. So that's what we call in vitro fertilization. In vitro is outside fertilization. We fertilize the egg outside the womb. We also do this if, for example, the mother cannot born baby. Some females, unfortunately, cannot carry a baby because it will put their health at risk and they could die. So one of the things that we do is we fertilize their egg outside of their womb, and then we can use another female. They have done this. For example, some of the grandmothers, they can carry the baby for their daughters. So that's what we call test tube babies. So we have been doing this. We can specifically choose an egg cell that we want or a sperm cell that we want. Cloning, we have done cloning. We have a cloned lamb. This is the first cloned organism that we have. She's called Dolly. We have we have taken a DNA from that lamb and then we create a new lamb that is identical to the original lamb. So cloning. We have done this successfully actually. So we have been doing genetic engineering for many years now. Only today, when we talk about genetic engineering, we're more specifically now talking about recombinant DNA technology. Previously, when we talk about genetic engineering, we talk everything that has anything to do with influencing and manipulating genes. But nowadays, when we talk about genetic engineering, we are more talking about the recombinant DNA technology that I just discussed about. And continuing on with historical developments, let's learn some names and people that are behind the development of genetic engineering. First is Werner Aber. Werner Aber is a Swiss microbiologist, and he's the one who discovered restriction enzymes. If you guys remember restriction enzymes, we just discussed this is the one that splices the plasmid and splices or cuts the DNA into multiple pieces, into 1,000 pieces, which is necessary for recombinant DNA technology. This is his picture right here. And then we have Hamilton O. Smith. So Hamilton O. Smith took Werner's discovery and improved upon it by creating a type 2 restriction enzyme. The difference is the type 1 restriction enzymes or the original restriction enzymes, it cuts the DNA into random pieces. But for type 2 restriction enzymes, we can now cleave or cut specific sites in the DNA. We can choose which section of DNA we want to cut. So this is more precise restriction enzymes. So here he is a American microbiologist. Next we have Daniel Nathans. Daniel Nathans took Smith's work and then said, we can use these restriction enzymes in our study of DNA, of our in our study of genetics. This is picture, and he improved the technique of DNA recombination in 1970 and 71. And lastly, the two people that basically pioneered the study of genetic engineering based on recombination in 1973 is Stanley and Cohen. This is picture, and Herbert Boyer. Here's his picture right here. So they cut the DNA into fragments and then rejoin them, basically creating the whole process of recombinant DNA technology. So these are the important people that are behind the development of our recombinant DNA technology. And lastly, let's talk about some controversy and ethical issues. So we've talked about a lot of great and good things about DNA recombinant technology and genetic engineering. But of course, this comes without saying that it's perfect. A lot of the things that is happening can be a controversy and can be an ethical issues. One of the things is if we are able to create new microorganisms, these microorganisms are patentable. When we say patent, it means the person who created these new organisms has a sole propriety or sole ownership of that microorganisms. And if these new microorganisms happen to be very useful in our lives, they can exploit it for money. So that's one of the ethical issues. Another ethical issues is if we create these new organisms, we do not know if they won't have a devastating or very dangerous long-lasting effect in the ecosystem. What if we create these microorganisms and in the beginning it's doing great and then suddenly it evolve into something very dangerous that can destroy our food, our crops, and can destroy other bacteria. We do not know yet. So because we don't have, because this is something new, we do not have yet a long-term study of these new microorganisms. They can be devastating in the long run or very dangerous in the long run because in the end, instead of evolving by themselves, they're created by humans, and humans are, because we're humans, we're made to err sometimes. So we are made to make mistakes sometimes. And because of that, this becomes a significant concern in our crops, in our health, and in the environmental health. And lastly, we can actually, another issue for ethical issue is exploitation on the use of, for example, the DNA tool. We can, for example, increase the intelligence and beauty of a baby. So there's a term that we used previously, it's called the designer baby. Because of CRISPR Cas9 and other gene editing tools, maybe in the future we can create the perfect baby. We can create a tall one, a beautiful one, basically making this way we call it a designer baby. We can pick and choose which traits our babies will want. And that is like playing God. You can think of it that way. You may think it's good, you may think it's bad, but it's an ethical issue. Because science in the end does not answer what is right or wrong. That is the job of morality. That's the job of morality. And science usually does not deal with an issue of morality. That's why science, we just want to advance, but we have to check whether what we're doing is right or wrong. So that's some of the ethical issues that we have with gene editing tool, and that's all our discussion about genetic engineering for today. If you guys have any question, do not hesitate to message me, and may you guys have a great day.